So welcome back in the another video tutorial from microscopy. So in this microscopy video lecture, today we will going to discuss about the last kind of microscopy, that is our scanning electron microscopy. So why we have developed the electron microscopy? Electron microscopy was developed to increase the magnification, right, along with the resolution, right. That is why we have chosen the electron as the illumination source, because electron have a shorter wavelength than the visible light, right. So under such circumstances, in our previous video we have discussed about transmission electron microscopy, but there was a problem that I have told you, that we are spending near about crores of money for transmission electron microscopy, but still we are getting what? Still we are getting a 2D image, right. So that is why, and that is the need why scanning electron microscopy, or SEM, which is our today's topic of discussion, was developed. Okay, because we all know there is a sentence that necessity is the mother of invention, right. And under such circumstances, the necessity was to see the three-dimensional image, right. So that is why we have constructed the SEM. So you may now ask that yes, we can use our confocal microscopy to see 3D image, right, but confocal microscopy cannot give you that amount of magnification and resolution that SEM can give you. Okay, so that is why that is what the difference between SEM and confocal microscopy is. Okay, yes, that there is no doubt that confocal microscopy can give you 3D image, but the magnification and the resolution will be not that amount that you can get when you will using SEM. Okay, so scanning electron microscopy, what can you understand from the full form of the SEM? That we are using electron to scan the specimen, isn't it? So that is the idea. Here we will going to use electron to scan the specimen. Okay, we will scan various part of the specimen, or each and every part of the specimen one by one, and then with the help of computer software, as you can see that here computers are attached, computer software, we will compile them, and as a result we will get the entire 3D image of the specimen. Okay, so now it is clear that SEM will give us a good magnification, good resolution along with 3D image, right. And here we are using electron to scan our specimen. Okay, so now it's time to learn about the working principle of SEM. So this is a GIF that I have taken from internet. I will going to provide the link of this GIF also in the description box. So we will use these GIFs to understand the principle of the SEM. So first thing, the instrument of scanning electron microscopy consists of two parts. One is specimen chamber, another one is column. As you can see, there is a high vacuum pump present. Okay, that will maintain the entire environment inside the instrument free of air or any kind of dust particle. Okay, otherwise it will create a problem during the analysis. Okay, and you can also see that we can move our specimen stage. Okay, and there will be also raster scan generator that I will going to talk about. This raster scan generator will help us to deflect the electron. Okay, and with the help of those deflecting electron we will going to scan our specimen. So the first instrument is the thermionic cathode. Okay, so when we will give a high energy to thermionic cathode, it will release electrons. Okay, high energy near about 20 kilovolt. Okay, so now after thermionic cathode there is a anode, okay, and both the anode and the thermionic cathode are attached with positively charged electrode and negatively charged electrode, okay, and as a result it will create a electrical field in between the thermionic cathode and the anode, okay, and this electrical field will accelerate electrons towards the electromagnetic lens, okay. You can thought this electromagnetic lens as a condenser, okay. This electromagnetic lens will condense the beam of electron towards the specimen, okay, and after the electromagnetic lens there will be also electromagnetic deflection, which is attached with a raster scan generator, and that is the raster scan generator which will allow us to deflect the electron. Let's say in that GIF the electron is falling on that part of the specimen, right? So we can use the raster scan generator to change the direction of the specimen, okay, just like that. And that's how, by using the raster scan generator and by deflecting the electron, we can scan the entire specimen, okay. So now after that the electron will hit the specimen, okay. So now there is a special kind of phenomena that is going on. So another important thing, that up to that part the electron that we are using and the electron which is hitting the specimen is known as primary electron, okay. Again, the electron that is coming from the thermionic cathode, then anode, then electromagnetic lens, then electromagnetic deflection, and then ultimately it is hitting the specimen is known as primary electron, okay. So now I think you know that specimen may consist of various molecules, right, and molecules should have atoms, right. So there is no doubt that the electron will fall on the atoms or will hit the atoms, okay. Now let's discuss what will happen. So as you can see this picture, when primary electron will hit the atom, it will knock out one electron from the inner shell, okay, and this knockout electron will be known as secondary electron. Now there is a problem. So another important thing, this secondary electron we will going to detect with the help of AC detector. This AC detector is secondary electron detector. I am going to discuss it on GIF. Before that I want to mention another important thing, that as the primary electron causes the knockout of one electron from the inner shell, so obviously there is a free space for one electron in the inner shell, okay, and that makes the atom unstable, okay. Under such circumstances, the one electron from the outer shell will jump towards the or in the inner shell, okay, as you can see here. And after that jump, because remember the energy of the outer shell will always be higher than the energy of the inner shell, okay, and this energy will be emitted by the electron during that jumping, okay, and this emitted energy will be of specific wavelength for specific type of compound, okay. That means, suppose let's say we are visualizing something that have, let's say we are visualizing or our electron, that is primary electron, hit a cell on its nucleus, okay. So under such circumstances the energy which will emit have a specific characteristics wavelength, okay, and that will be detected by X-ray detector, which is a additional detector present in scanning electron microscopy, okay. That means two type of detector present in scanning electron microscopy. One is the scanning electron detector or the secondary electron detector. And what is secondary electron? The electron that is knocked out by the primary electron, right? And it is where it is situated? It is situated in the inner shell, right? So now, and another detector will be the X-ray detector, which will detect the energy emitted by the electron during the jumping from the outer shell to the inner shell. Okay, so it is clear that due to the jumping of the electron, now inner shell is in its normal state. Okay, but there is a problem that is situated, or that is created in the L shell, right? So to solve this problem, one electron from the outer shell, that is the M shell, will jump towards the L shell. Okay, and here again there will be a emission of energy, which is also of particular wavelength. Okay, and this energy of particular wavelengths will be detected by the X-ray detector. So that is what is happening. So as you can see that there is a AC detector. AC detector means secondary electron detector, which will detect the secondary electrons. Okay, and another important thing to observe, that in the AC detector there is a red marked portion which is known as positively charged grid. Okay, this positively charged grid will attract all the negatively charged electron towards the detector. Okay, and after that the detector will transfer those information in computer, and computer will start generating the image. Okay, then as you can see here, the electron is deflecting with the help of raster scan generator. This raster scan generator will help us to create the entire image. Okay, and there will be another detector which is present just like this point, that is the X-ray detector. With this X-ray detector will detect the wavelength of the energy emitted by the electron. So that is the entire idea. So again, a quick recap, that the entire instrument of scanning electron microscopy is of two parts. One is specimen chamber, another one is column. Okay, and there is a vacuum pump present. This vacuum pump maintain the inside environment air free and dust free. Okay, and here the electron source is thermionic cathode. You can also use hot tungsten filament or lanthanum hexaboride. Okay, so whenever you will give a high energy electron, whenever you apply a high energy which will be of 20 kilovolt, the thermionic cathode will start releasing or producing electrons. Okay, these electrons will move towards the anode, and this anode is attached with positively charged electrode. Also the thermionic cathode is also attached with the negatively charged electrode. Okay, and there will be a generation of strong electrical field in between the cathode and anode. Okay, this electrical field will accelerate the electron towards the electromagnetic lens. Okay, and this electromagnetic lens is kind of a condenser. This electromagnetic lens will focus the beam of electron towards the specimen. Okay, and there is also a electromagnetic deflection which is associated with raster scan generator. This raster scan generator will allow the deflection of the electron. Okay, and this deflection of the electron will help us to scan each and every part of the specimen. Okay, and what is going on? So this electron is known as primary electron, I have told you. So when this electron will hit the specimen, it is obvious that it is hitting the atom of the molecule present in the specimen. Okay, and by hitting the atom, it will causes the knockout of one electron from the inner shell. Okay, and this knockout electron is what we are detecting with the help of this secondary electron detector. Okay, and for regaining the stability of the atom, the electrons which are present in the outer shell of the atom emitting energy, and those emitted energy are of specific wavelength and which is detected by a X-ray detector. Okay, and now this secondary electron detector will collect all the information and transfer it to the computer, and computer will compile each and every information, okay, and create a 3D image of the entire specimen. Okay, so that is the entire idea of scanning electron microscopy. So another important thing, sorry, that it is possible that some part of the specimen have a high density of molecules, okay, so and some part of the specimen will have a lower density of molecules, and similarly some part of the specimen should lack any kind of molecules. For example, let's consider, if we are considering a cell as a specimen, then the cytoplasm will have the lower dense part, right? Similarly, nucleus will be the most dense one, right? So whenever electron will hit the denser part of the specimen, it will create more secondary electron. It is obvious, right? There is no doubt about this, because more denser part of the specimen means more molecules, and more molecules means more atom, and more atom means more chance of knocking out of electron, right? And that is our secondary electron, right? So as a result, the denser part of the specimen will be appear bright, okay, and the part which has a middle density which will appear, that will appear as a gray color. Again I am repeating, for example, let's say we are considering cell as our sample, okay, so the nucleus will appear in bright color, okay. On the other hand, let's say mitochondria will appear gray in color, okay, and the cytoplasm part will appear black in color, because cytoplasm here of lowest density, right? And lowest density means obviously lowest amount of molecules, and lowest amount of molecule means lowest amount of atoms, and lowest amount of atom means lowest amount of knocked out electrons, right? So that is important thing to remember, that the denser part of the specimen will appear bright, middle denser part of the specimen will appear gray, and the lowest denser part of the specimen will appear black, okay. So that is how the entire scanning electron microscopy works fantastically to give us a three-dimensional picture of our specimen, okay. So now it is time to understand about the advantages and disadvantages. So advantages is obviously now we can visualize them in 3D, okay, and disadvantages is we cannot still visualize live image of the specimen, okay, because remember, I think you can remember that I have conducted a lecture on the sample preparation of the electron for electron microscopy, right? And where we have discussed about the dehydration step, right? So what do you think, that if you dehydrate a live specimen, can it will survive? No. That is the reason why both transmission electron microscopy and scanning electron microscopy cannot give you image of the live specimen. For visualizing live specimen, you have to rely on phase contrast microscopy, our dark field microscopy stuff, okay. And another important disadvantages is the image is still black and white. In electron microscopy, you cannot get image in color, okay. So that is the concept of scanning electron microscopy. So what are the different type of field where we can use scanning electron microscopy? We can use scanning electron microscopy for the three-dimensional analysis of the tissue. Okay, not only the biological field but also in non-biological field, a geologist can use scanning electron microscopy to for detailed study of crystals, okay, and or crystalline structures, okay, and along with insect spores and other microbes and variable cellular components can also be visualized, and it can also be used in industries for analysis of the product, and also the surface composition of the component can be visualized with the help of this scanning electron microscopy. Okay, so we are using it mainly for investigation of the virus structure. We are using it for analysis of the spores, other microbes or cellular microbes, component visualization, along with also geologist uses scanning electron microscopy to visualize crystalline structures, and in industries same is widely used for analyzing surface composition of the component and product. Okay, so that is the concept of scanning electron microscopy. So I hope this video will be helpful to you to understand the basic component, basic concept of the scanning electron microscopy. So thank you for listening to this class.